My name is Tom Bollenbach, and I'm the co-founder and chief technology officer of the Advanced Regenerative Manufacturing Institute, or ARMI. In this talk, I'll be summarizing the activities of ARMI's BioFabUSA program, which is providing an accelerated path to scalable, consistent, cost-effective manufacturing of cells, tissues, and organs. You might not have heard of ARMI or BioFabUSA, as we really haven't been a fixture at Meeting on the Mesa in the past. However, whether you're running or investing in a startup and struggling to implement manufacturing of your product, or you otherwise want to accelerate your path to commercial scale manufacturing, you're either already working with us or you should be working with us. And if you do represent one of the 170 plus organizations already working with us, thank you. If you've been following the history of tissue engineering, you'd know that although the field's been around for decades and that there's been a significant investment to the field, including nearly $3 billion in federal research and development funding between 2012 and 2014 alone, that these products have been tough to commercialize, in large part because scalable manufacturing has been a bottleneck. An example of a technician working in a manufacturing environment of today is shown in the upper left panel. The process is labor intensive, and so it isn't scalable unless you add more technicians. Process control is a challenge because we really don't know what to measure either in process or at product release in many cases. These tissues are also at risk of contamination because the trays are open and exposed to the ambient air repeatedly. And to mitigate that risk, the process is conducted in a clean room environment, which is extremely expensive to operate. The point here is that this is a scientist's version of manufacturing, probably because this is how the construct was fabricated in Eugene Bell's lab at MIT as far back as the late 1970s. To use an analogy, we need to think of manufacturing of cells, tissues, and organs less as an artisanal process like making grandma's chicken soup and more like they do at Campbell's. So having said that, whose job is it to address this manufacturing bottleneck? It shouldn't lie with the product developers. Their expertise is in biology and medicine, not engineering, automation, robotics, and quality systems. Over the last 4.5 years, it's become the collective responsibility of a large and growing consortium with highly diverse strengths across a number of scientific, engineering, data analytics, and regulatory science disciplines. In order to coordinate this consortium and to invest in solving these manufacturing challenges, ARMI, a 501c6 nonprofit based in the greater Boston area, was awarded an $80 million grant by the Department of Defense to stand up the BioFabUSA program, which is focused on advanced manufacturing, research, and development for cells, tissue engineered medical products, and organs. We launched BioFabUSA in the summer of 2017 and have more than 170 member organizations comprising, in order of abundance, small and medium-sized enterprises, large companies, including pharma and biopharma, academia, nonprofit organizations, which are all spread across the United States. We also partner with government agencies and have a public-private partnership agreement with CBER, which allows us to interact with FDA as a neutral third party to socialize new manufacturing concepts and technologies with them before product developers implement them. I want to emphasize that this is truly a team effort, and I'm not going to go through all of these members, but I do want to point out that our members include not only cell, tissue, and organ developers, but everyone that's required to tackle various challenges across the entire process and supply chain to make manufacturing of these living products a reality. This table compares where we are today with the collective vision of the future for cell, tissue, and organ manufacturing that was articulated by our member organizations and that's captured in our latest technology development roadmap. In the future, process unit operations will be simplified and integrated through partial or full automation. Labor typically comprises 30 to 50% of the cost of cell and tissue products. So there's a significant need to minimize headcount and minimize variability through automation. Operations will be conducted in controlled, non-classified spaces with environmentally controlled, closed manufacturing systems. In large part, the cost of operating a clean room is a result of energy consumption. ISO 5 spaces, where these operations are typically carried out, have 25 times the energy costs of a non-classified space. All of that contributes to the cost of the final product. Quality control specifications will be correlated to product efficacy through deep characterization and advanced data analytics, and the relationship between final product quality and manufacturing process parameters and material attributes will be well defined through robust in vitro characterization and advanced data analytics. And processes will be controlled at their most critical steps, and flexibility will be allowed where appropriate. The biopharmaceutical industry has already shown the benefits of quality by design. Companies report zero process atypicals and significant reduction in costs because of reduced need for final product testing. Processes will be monitored by non-destructive sensors like they are in other industries. Raw material characteristics will be defined and standardized. Most manufacturing processes we take in at the Institute still use animal serum as a component of culture media, something that the biopharmaceutical industry moved away from years ago. There isn't enough serum in the world to sustain scaled manufacturing of these products, and serum is also a huge source of process variability and a supply chain risk. And finally, we'll have more flexible logistics by design-based platforms for preservation and transport of products that either manages or eliminates the need for a cold chain. I want to point out that this isn't only a vision. We're currently working on collectively on more than 40 technology projects to enable this future state now across three major technical activities. Technology development, where we're building enabling technologies that complement existing process technologies, and building platform technologies for broad applicability so that they're process agnostic. We also focus on deep product and process characterization for quality by design, where we identify quality control specifications for complex living products. And finally, we conduct process development and integration of new technologies and automation in scalable, modular, automated, and closed manufacturing systems. And I'll dive into each of these a little bit more now. We have nearly 30 ongoing enabling technology development projects, which are driven by an industry needs roadmap. And some examples of these are shown here. We've invested with our members in closed, centralized culture control systems, in bioreactors with embedded sensors for non-destructive measurement of in-process critical quality attributes of cells and tissues, including not only pH, dissolved oxygen, and the ones we typically would measure, but also metabolites, lipids, and proteins, as well as adventitious agents in real time. We're taking on the challenge of developing chemically defined culture media and cryopreservation agents and chemically defined culture surface substrates for a variety of cell types through screening platforms in development by a number of our partners. And finally, we're taking on the challenge of accurately and reproducibly recellularizing whole organ scaffolds, starting with the heart, using AI-driven robotics and automation. The point here is that we're working on a number of platform technologies that will eventually be available to our members and later to the industry as a whole. In a moment, I'll describe the work we've been doing to close and automate several manufacturing processes. One of the biggest challenges we've seen with adapting various processes to automation is that there's a big gap in the amount of process data and final product characterization that have been done. Having sufficient characterization data are important because it lets us know the level of comparability between the final products generated manually and under automation, and then lets us know the level of comparability between products generated at increasing scale. We can't assume that simply automating the process will faithfully replicate what's done manually. That comparability hinges on a robust set of in-process and final product critical quality attributes, or CQAs, including potency, purity, and identity, which are also required to be developed for federal regulation. Historically, identification of these attributes has been largely guesswork. We're taking a systematic approach to identifying these CQAs by performing in-depth multi-omic, phenotypic, and mechanical characterization of final product, and then apply modern tools of data analytics like multivariate data analysis, machine learning, AI, and so on, to understand which subset of those characteristics correlate with preclinical or clinical data. It's that subset of quality attributes that are critical to efficacy in the patient that will become the QC specifications for potency, purity, and identity that we need. We also focus heavily on the identification of non-destructive measurements that can eventually be performed by a sensor. That means we also focus on deep characterization of conditioned culture media and eventually apply a sensor to that measurement in manufacturing. So it's not only non-destructive, but also inline and real time, or at least sampling at the appropriate frequency. The cornerstone of this approach is measurement, a lot of measurement. And for that reason, we launched the ARMI Deep Tissue Characterization Center, shown at lower left, which is a core facility that's widely available to address this exact challenge as you develop your process. Its main function is to conduct deep characterization of the intercellular and secreted proteome, lipidome, metabolome, and transcriptome, and to do phenotypic characterization of surface markers to test tissue and organ mechanical properties and to sample headspace gas. We're also now able to do leachables and extractables testing. I also mentioned technology integration, in which we close processes and adapt them to automation. The easy thing to do would be to envision a system of robotics that takes flasks and performs unit operations like aspirating and refilling culture media the way a technician would. But a robotics approach doesn't lend itself to the incorporation of sensors or easily to scale because we can't take advantage of culture systems any larger than typical T-flasks. So then we consider automation. So what I show here is an example of an automated manufacturing line that we built at the Institute to run a process developed by Stell Technologies to manufacture ligaments for ACL repair. The starting material for the ligaments are bone marrow-derived mesenchymal cells that generate a hybrid of bone and ligament in a 45-day process. An example of the final product is shown at right. This prototype system automates each step of the manual process, which it's compared to along the top row, and is capable of running autonomously without operator intervention after the frozen vial is introduced to the system. Because the system is modular, we can introduce various types of technologies at various scales at each step. One of the features of this system is its ability to log data from both the automation equipment and from various types of measurement technologies, including sensors. Quite a bit of the work we're doing at the moment focuses on the development of sensor technologies to complement the multi-omic characterization I described in the Deep Tissue Characterization Center. This prototype line will remain in place as a test bench for new technologies that I described earlier. And we currently have a queue of new technologies that are waiting to be integrated. And we're now adapting 10 additional processes to automation and building additional tissue foundry systems to support them. These lines will be producing skeletal muscle, bone, pancreatic islets, retina, heart, skin, and several others. This approach to scalable, modular, automated, and closed manufacturing has really allowed us to think critically about facilities design and the idea of moving the industry toward manufacturing in controlled, unclassified spaces. As a first step toward that future state, we are completing construction on a 30,000 square foot facility that'll combine manufacturing suites certifiable to ISO 8 for closed manufacturing with an ISO 7 cell banking suite to accommodate partially open unit operations, all to support process development and early phase clinical manufacturing. In late 2021, we'll launch the new facility as BioFabFoundries, which will co-locate our process development, automation, deep cell and tissue characterization teams in a building adjacent to our regulatory, quality, and commercialization arm known as BioFab Consulting here on our Manchester, New Hampshire campus. This new facility will be available for both DoD-sponsored process automation projects that I described earlier and traditional CDMO type activities that we would love to engage with you in. So I summarized an overview of ARMI and the BioFabUSA program and its technology programs, including technology development, deep tissue and cell characterization, and process development and technology integration. If you're keen to participate in technology development, characterization, or automation, or have technologies that can be applied to enable manufacturing, then we're eager to hear from you. Contact us at info@armiusa.org or on our website at www.armiusa.org. Interacting with us can be done through our normal project call mechanism, which will be listed on our website, in which we co-invest in projects with federal funds, or it can be done through more traditional CDMO services or through BioFab Consulting, depending on your needs. Thank you.